Welcome everybody. Thank you for coming. I think we've got a number of people online as well. So as you know, this is part of our Scott lecture series, which we're super delighted to have back in person in 3 dimensions, as well as hosting people online who are watching, for those who prefer to do it that way. And we're really excited today to have today's speaker, Soumya Raychaudhuri. Let me see if I can make that go. One sec. There we go. And so, as you know, this is, there we go. This lecture series is named for Richard A. Scott. It was endowed by Anne Scott and Dr. Scott's family members. And this is a picture of them from when they were meeting President Bush in Prague many years ago. And they were very active in the Czech community and in Czech in general. So, and a picture of Dr. Scott at a young age. He was an eminent pathologist and really made a lot of important contributions. But I would like to focus on Mrs. Scott. Make sure I didn't miss anything there. As some of you know, we recently lost Mrs. Scott. She was able to make it for the last in-person Scott lecture that we had not that long ago. Quite valiantly showed up and was asking questions and very, very engaged, but she did pass away in late March. This is just really a tribute to Mrs. Scott. She was such a lover of science and investigation and all things genetics, and she really carried that with her and was asking all those great questions throughout her entire life. And so this is really just a true tribute to her. And I know I miss her, and I know her family members miss her, but it was just such a pleasure to get to know her. And so our speaker today, Dr. Raychaudhuri, is coming to us from Harvard Medical School. And let me just tell you a little bit about his background. So he received his undergraduate degrees from the State University at Buffalo, and with majors in biophysics and mathematics and minors in chemistry and physics, so very well educated. And then went to Stanford, where he did both his MD and PhD degrees, including a PhD in biomedical informatics, where he worked with Russ Altman, and then completed his MD in 2004. And from there, he that's when you moved to the Brigham and moved to Boston and did his residency in internal medicine and subsequent training in rheumatology. And from there, he's just really had a marvelous career where he will tell us some of his really interesting work today. I just want to point out some of his work has been acknowledged through external awards. So he's a member of the American Society of Clinical Investigation since 2016, received a Doris Duke Award, and importantly received the Henry Kunkel Award, which was a Young Investigator Award from Rheumatology Society. And I trained with Lou Kunkel, so that's kind of very cool to hear, the Henry Kunkel Award. We're actually going through some of the, JCI is having its 100th anniversary next year, and so we actually have some of Henry Kunkel's papers that will feature in that. So he's the Coblyn and Brenner Chair and a professor now at Harvard Medical School, and he is a member of the Broad Institute as well. And so with that, I'm so delighted that that Dr. Raychaudhuri could join us today, where he's going to talk about genetic mechanisms of autoimmune diseases, a very important topic. And so with that, we will get started. And I don't know if we're switching computers. Is that what we're doing?
Well, thank you so much for that really nice introduction, and thank you so much for having me here. It's a real treat for me to be here in Northwestern. It's the first time I've come and spoken here, but it's an institution that's really important to my family. In 1965, my father came here to pursue his graduate work, so You know, he's the first member of our family to come here. So, and this is him in Evanston. I guess he would have been a contemporary of Dr. Scott's. They must have overlapped as students. So I'm going to talk today about autoimmune diseases, and this whole set of diseases are conditions that occur when your immune system is activated, usually by an autoantigen, a self-antigen that results in your immune system attacking one of your joints or one of your organ systems. And some of these are very organ system specific, like multiple sclerosis or thyroiditis, and other diseases are much more systemic, such as systemic lupus erythematosus, which can affect a wide range of different tissues. This is a children's book about a person's experience with type 1 diabetes. This young girl learned to inject herself with insulin when she was small, and it gave her the courage to go on to do many great things in life. This book is by Soumya Raychaudhuri. She had type 1 diabetes. Another disease that I think a lot about, and I spend most of my career thinking about, is rheumatoid arthritis. This is a disease that was first described in the year 1800 or in year 8 of the French Republic. Graduate students would be inspired to know that this was a student dissertation describing rheumatoid arthritis with 9 cases, which, you know, in this day and age, is very hard to get anything published with 9 cases, much less name a disease. The student noticed that this form of arthritis was predominantly affecting women, had a chronic course, affected multiple joints, which made it stand out from gout, which was then the common form of arthritis. Rheumatoid arthritis affects up to 1% of the population worldwide. It causes a symmetric destructive arthritis of the hands. Multiple joints can be affected, particularly besides the hands, including the neck, knees, ankles, wrists, shoulders. There can also be effects on other organ systems, such as the lungs or the blood vessels, causing a vasculitis. On the right, this is sort of the typical picture of rheumatoid arthritis. On the left, you have a healthy joint, in this case, a knee, and you can see the cartilage there between the two surfaces. And surrounding the cartilage is this thin tissue called the synovium, which is normally barely visible. And when the immune system attacks it, it becomes engorged and inflamed, and that leads to the destruction of the cartilage, destruction of the bone, and that's what leads to the disability and joint destruction that accompanies this condition. This is a person who had rheumatoid arthritis. This is Pierre-Auguste Renoir. He, in the late stages of his life, his hands became unusable because of rheumatoid arthritis. It didn't stop him from making paintings like the self-portrait on the right. These paintings were made with a brush strapped to his wrist. Like many autoimmune diseases, rheumatoid arthritis has a compelling genetic component to it. In the general population, about 1 in 200 individuals have RA. It's about 0.5% of the population. But if you have an affected first-degree relative, your risk goes up 3- or 4-fold. So for example, if you had an affected parent or an affected sibling, your risk is increased. I always find it interesting that if you have an affected spouse, you actually are at a 17% higher risk of the disease, probably reflecting the impact of a shared environment. So for example, smoking and pollutants are thought to be associated with RA. Similar numbers for type 1 diabetes, and you can run through a lot of different autoimmune diseases in this way. You can see it's a little bit different than a Mendelian disease or what we think of as traditionally a genetic disease, in that your risk goes up 8-10-fold if you have a first-degree relative with whom you share half your genome with. Rheumatoid arthritis and autoimmune diseases in general have really been a productive area for drug development over the last couple of decades. You can see in the 1950s, 1960s, 1970s, we were doing things like injecting Oranophen. Oranophen is gold. It wasn't very effective. And then sort of into the 80s and 90s, we got very good at using conventional disease-modifying rheumatic drugs. And then, of course, we moved into the targeted therapeutic era, where a broad range of medications became available that target individual cytokines that really have changed the long-term outcomes for our patients. But it continues to be important to understand how this disease and other autoimmune diseases work. The therapies have improved, but there's still gaps. One of the difficulties with this condition that any patient with rheumatoid arthritis will tell you about is that it takes us a long time to figure out what the right therapy for the right patient is. And there's a real need for novel therapeutics, since immunosuppression, which is what all those medications do, doesn't consistently lead to remission. And of course, there's still no cure. We've been using genetic studies to understand the mechanisms that drive RA susceptibility. Our latest study is here. It's a worldwide collaboration. You can see all of the different cohorts and institutions that have played a role here. This study involved 35,000 cases, 240,000 controls, and it's contributed to a mounting list of individual SNP associations throughout the genome that are driving RA risk. You can see prior to 2003, there was almost nothing. There was an MHC association. And then with each year that's gone by, our list of alleles have been expanding, and now we have somewhere on the order of 150 risk alleles that are known to drive RA susceptibility. But the risk across all loci aren't equal. So this is a typical plot of a genome-wide association study. Some people call this a Manhattan plot. You can imagine this is a skyline, and each point maybe reflects how statistically significant that change in the genome, that SNP, is associated with. So usually we're plotting, of course, the negative log p-value. But you can see Manhattan is like a poor representation. There's actually like this big central tower. So it's probably more like Dubai. And this, of course, is the major histocompatibility complex, or the MHC locus, which plays not only an important role in autoimmune diseases, but plays an important role in many, many, many conditions. And this is a slide courtesy of Mary Carrington, which just shows how many different conditions have been linked to this really important locus. Not just autoimmune diseases, but also cancer, viral infections, neuropsychiatric conditions, and so on and so on. And we'll be diving a little bit into this locus later in the talk. One of the resounding themes of the work that we've done and others have done in terms of understanding the genetic basis of autoimmune diseases is that T cells, unsurprisingly perhaps, are really important and are strongly implicated in most autoimmune diseases. The HLA locus, of course, is intrinsic to the presentation of a self-antigen to the T cell receptor, triggering the immune response. But outside of the HLA, there are about, if you start looking at those alleles, what you'll see is that about 10% of them alter a coding sequence, but 90% of them seem to be non-coding alleles. But they're disproportionately close to T cell genes, and they're disproportionately present in regulatory regions that are T cell specific. I'm going to be talking in the rest of the talk today about the relationship of these alleles and T cell function and T cell dysfunction and T cell identity. So when we think of T cell identity, there's really two aspects that I think about. The first is the recognition of a foreign antigen on the left, and the second on the right is the specific T cell states that a T cell takes on. So T cells are stimulated by HLA and antigen. So the HLA, or the major histocompatibility complex, in this case, for most of the diseases I think about, the class 2 MHC, presents an antigen to the CD4 T cell, which results in an antigen-specific interaction, leading to a state change in the T cell as it gets activated. And a lot of times we think of that activation as moving from a naive T cell into some sort of memory effector T cell state. And there's a whole bunch of canonical T cell states. Most of these aren't controversial, but some of them might be. Th1, Th2 T cell states, for example, are some of the typical ones one might think about. So when we think of the identity of a T cell, the T cell receptor is really important because the T cell receptor is what tells a T cell which specific antigen it should recognize. The TCR is randomly generated. The CDR3 region, which I'm going to be talking a lot about, recognizes specific autoantigens or antigens. It determines the antigen that a T cell will respond to. And thymic selection selects T cells based on the TCR. So negative selection happens in the thymus to remove T cells that are recognizing self-antigens, but positive selection happens to make sure that the TCR that's being selected is able to recognize the MHC in general and is able to sort of trigger some kind of a response. Another form of a T cell's identity is the transcriptional state. The transcriptional state, can be measured in multiple ways. We measure it a lot by RNA-seq, but it can also be picked up by surface protein markers. After activation of T cells become naive to memory T cell states, and of course they take on these different states that I told you about. And traditionally we recognize these states with surface markers, but increasingly we're using single cell analyses to look at RNA-seq profiles of these T cell states to understand their profile. Let me start by talking a little bit about the right side here about transcriptional states of T cells. And so for the first part of the talk, I'm going to talk about defining T cell states and their effects on gene regulation. And then in the second part of the talk, I'm going to talk about how HLA alleles influence T cell receptor composition. And finally, I'm going to talk about how T cell receptor sequences might be influencing T cell states. So for a lot of these associations that come out of the studies, this is what your typical locus looks like. You've got a whole bunch of SNPs, and you can see that there's a big red row of SNPs that are strongly associated with the phenotype. And for a given locus, it's really important for us to understand which SNP is causal, which ones are kind of just correlated, which ones are linked. It's important for us to understand the functional effect of the SNP, and of course we want to know ultimately what the causal gene is. And also, since we're talking about gene regulation, it's important for us to understand what the causal cell state is, because there are different mechanisms regulating genes in different circumstances. And I think of this problem a little bit like a Jenga tower. And those of you who think about non-coding alleles think a lot about this, because you sort of need to figure all of these things out simultaneously. So when we started looking at these alleles for RA and other diseases, we started thinking about these non-coding alleles, and we wanted to ask a simple question: do they influence gene regulation? And the first clue, and that was important for me, was to start looking and asking, are these alleles in regulatory regions? And we, of course, were using histone marks. And at that time, histone mark data was becoming widely available. And we could simply ask, are these SNPs falling in regions that are enriched for specific histone marks and specific cell states? And we recognized that the effect of these alleles might be cell state specific. So this study that we published in 2013 was one of our early clues that RA risk alleles were highly enriched of all the different cell states that we looked at, most enriched in CD4 T cell memory subset. So that was really interesting. And about a year later, Kyle Farr, Alex Marson, and colleagues looked across a wide range of diseases, including autoimmune diseases, and they started looking at histone marks and asking similar questions. And what you can see is that CD4 T cell states for a broad range of autoimmune diseases is again highly enriched. So it seemed like paying attention to these regulatory elements was really important. And one of the key mechanisms that we started to think about was expression quantitative trait loci, which I learned for the first time by reading a paper by Barbara Stranger many years ago. The idea, of course, is that you have a SNP, it's sitting in a regulatory region. Perhaps it's altering the way a transcription factor is binding that SNP, because you bind A better than a T. And maybe that has a predictable effect on gene expression. So this particular gene is the BLK gene. It's an RA SNP. Depending on whether you have 1, 2, or 3 copies of the CC, the C genotype, you have a lower level of expression of that BLK gene. So that's pretty encouraging. And this is the sort of thing that we want to typically see that makes us very happy. So at the top here, I'm showing all the SNPs and their association with BLK expression. And you can see that there's like a red SNP there. That's your rheumatoid arthritis SNP. It's one of the most strongly associated SNPs with BLK expression. And we would say it co-localizes. If you condition on that SNP, you see that all of the signal goes away. So that SNP fully explains the expression of BLK and T cells. So this is a positive example. You've got a SNP. It's associated with expression of BLK. It fully explains that association, the disease SNP. So you would probably conclude that the RA risk allele is working by modulating expression of BLK. But most of the time, things are not so good. About 50% of the autoimmune risk alleles that we've looked at are within T cell regulatory regions, but only 10 to 20% of EQTLs co-localize with T cell EQTLs in that way that I showed you. So what are all these other risk alleles doing? My colleague Shamil Sunyav looked at this problem in a really nice study where he looked at 139 genes that he felt were causal for different phenotypes. He knew they were causal because coding alleles that disrupt those genes in a convincing way drive a phenocopy of the complex disease. Only about 7% of the genes that they looked at did he notice that there was this really nice co-localization pattern. This is a SNP that we've spent a lot of time thinking about over the years. This is a SNP that's associated with rheumatoid arthritis and type 1 diabetes. It's within the CD28 CTLA4 locus. So if you have an A versus a C allele, when you have the C allele, you can see that you get a positive EMSA signal. So this indicates that there's a T cell extract that's binding the C allele that's not binding the A allele. On the right, we've done a luciferase assay. So you can see that the A allele has luciferase activity, indicating that it's got regulatory potential. If you've got the C allele, it's got even more signal. So the C allele is clearly doing something. And then if you do a promoter capture high C analysis, this is from public data, you see that that SNP is sitting in a region which communicates with the CTLA4 promoter. So you would expect that the SNP is somehow altering a regulatory element, which is interacting with the CTLA4 gene promoter. So it should have an effect on gene expression, but actually you don't see any association with CTLA4 expression. So this is a problem that's always troubled us. It got us to think about the question, like, does functional state matter? Is it possible that the effect of a SNP on gene expression is not just cell type specific, but also cell state dependent? Perhaps an allele that's influencing gene expression in a naive cell won't be doing so in a memory cell, or perhaps if it's working in Th17 T cells, it won't be doing so in other memory T cell states. So we thought that perhaps regulatory regions may be active within specific T cell states, and it would be valuable to understand how these alleles work and what that cell state context dependency is. So we were, of course, thinking about single cell data, which was coming online right as we were starting to think about this problem in a serious way. And we started to wonder if we had a map of all of the T cells, perhaps we would find that there were some cell states where there really is no association between genotype and expression, but perhaps there's some states where there was a strong association between genotype and expression. The goal here was to use single cell data to look at EQTLs to understand if they have the largest effect on expression in one cell state. And the particular strategy that we took was to look for EQTLs that were dynamic across cell states, looking at data in vivo. And it was important for us as we did this to do multimodal phenotyping of T cells, where we could see both the RNA level data that I was telling you about, but also to see surface marker protein data, because so much of what we understand of T cell states traditionally has come from these surface marker stains. So some of the challenges that we thought a lot about as we approached this problem was that memory T cells are not discrete, but they're a continuum. As we've looked at single cell data, we don't think that T cell states are quite as distinct as we like to think of them. Rather, they sort of bleed into each other. And we needed to think about how do we model EQTLs given the sparsity of single cell data. And of course, we started to think about different models than the standard models. And again, as I mentioned, and it's no secret that when we started this project, single cell data and its throughput was really blowing up. And that meant that we could start taking a whole cohort of individuals, applying this technology and doing these kinds of studies. So this project was done by, was led by Aparna Nathan, who was then a graduate student in my lab. Now she's a lecturer in biomedical informatics. We assayed 250 individuals. We assayed their PBMCs. It was a Peruvian cohort, and we assayed 500,000 memory T cells. And we obtained RNA and surface marker data jointly. And we jointly modeled their the RNA and protein markers together using canonical correlation analysis. And I'll just talk about that for a second. So we used site-seq data. So we had antibodies that had an oligonucleotide label. We stained the cells, and then when we sequence for the RNA, we're able to also get information on those oligonucleotide labels. So that tells us how much of a particular antibody is sticking to the T cell. We designed a custom panel which captured T cell function and also included antibodies that gave us really good signal. And the particular method that we used here was canonical correlation analysis. So the data was what's on the left. We have both RNA and protein data. And then we use this method from Harold Hotelling, which he described in 1936 called CCA, which has become really, really popular in the single cell timeframe and has found a resurgence during that time. But the basic idea is that you've got a matrix of cells and genes at the top, and you also have a matrix of cells and proteins. And you're looking for linear combinations of genes that are highly correlated with linear combinations of proteins. So it's a lot like principal components analysis, but it's forcing these things to be correlated with each other. So, so this is what our data look like. So we had data on 500,000 cells, and we had these different canonical variants that captured sort of different groups of genes and different groups of proteins. And the typical thing you might do is you might take all these canonical variants and apply clustering. And then what you would end up with is a picture like the one we have up at the top, where you have 31 different clusters of T cells. And of course, you can look at individual genes and individual proteins and feel very good that you're seeing the kinds of T cells that we know and love. But again, we want to get at this question here, are alleles associated with gene expression? And again, we want to think about those canonical variants. So the first question we asked is, do these CVs have some sort of biology behind them? And indeed, as we did pathway analysis and such things, we saw that they did. So the first CV, for example, CV1, you can see is capturing the left to right gradient. And the furthest right cells are the highest expressors of CD8, coenzyme B, an innateness gene set. So we think that this was capturing sort of cytotoxicity. CV2, which sort of picks up on this north-south axis, you can see the cells on the south side. They're the ones that are most lit up. They have the most positive CV2 signal. They're also the highest expressors of FOXP3. This is picking up on a T regulatory phenotype. So different biological functions are being picked up by these different CVs. So now we can go through and simply annotate all of these individual CVs and assign specific functions to them. And now when we think about genetic variation and its effect on expression, we can actually, rather than modeling, doing 31 different models and all of these different sparse small clusters, we simply use the CVs and say, does the EQTL correlate with these CVs? And so this was the model we used. This is an interaction model. We used a Poisson mixed effects model, which allows us to take into account really sparse data. So rather than using regular linear regression, which doesn't work given the sparsity of the data here. And the first model is like your typical EQTL model, where expression is associated with genotype, and your beta naught here is that effect. And now what we can do is we can think about the fact that these cells have different cell states. We can model differential expression by including a cell state term as indicated here. And then finally, we can model an interaction with cell state. So now you can see we've got a beta naught and a beta 1 term. And in this case, the beta 1 term tells you how the effect of genotype on expression is changing with different cell states. And every cell has a unique cell state variable. So we can actually define a specific genotype effect for each cell in this way. And then on the right, we can then have a picture like the one we have here, where you can get that cell type specific effect of the SNP on gene expression. So, so our strategy here was given the computational complexity of it, we started by identifying all the genes with an EQTL kind of across the board. So that was 6,500 genes. And then we used our model to ask which EQTLs get bigger or smaller with a change in cell state. That is, do the CVs describing T cell state interact with EQTL effect size? So this is one gene, this is the MAF gene. And you can see here that this, on the right side, the SNP is having a bigger effect on MAF expression. And then you can look in the lower third, middle third, and upper third of this. And you see if you take and group all the cells in the lower third, the association between the SNP and the gene expression is minimal. But on the far right, you can see that the SNP is really affecting gene expression pretty dramatically. What we found is that about a third of the T cell EQTLs that we looked at were indeed state dependent. 2/3 were not state dependent. And really interestingly, when we started looking at our autoimmune disease alleles, we found that more than half of them for many traits, and in the case of rheumatoid arthritis, 71% of them had state-dependent features. This was one of the most interesting ones for me. This is CTLA-4. So what you can see here on the left is that the SNP is influencing gene expression more dramatically for all these yellow cells. But if you look at where CTLA-4 is expressed, it's actually most highly expressed in regulatory T cells. So this is a situation where when the gene is highly expressed, the SNP doesn't really affect the expression of the gene. But when the SNP is lowly expressed, the SNP allows kind of leaky expression. So you have sort of an ectopic expression model. So people who have this particular SNP are more likely to express this regulatory gene in non-regulatory T cells. And this SNP is a protective SNP for autoimmune diseases. Just to give you a picture of some of the stuff we're thinking about now is we now want a platform for functional validation. So as we're finding these SNPs and as we think that they are having effects on gene expression in a context-dependent way, we want a platform to edit and introduce these SNPs to confirm the functional effect of these alleles. So our plan is to create these cell states in vivo, and then to edit simultaneously these cells. So it requires a platform where we can do RNA sequencing to capture cell states. We also, at the same time, need to do DNA editing to know if we've been successful in editing a cell or not. And of course, we need protein markers because we're talking about T cells. And given how heterogeneous the cell states are and how inefficient editing is, we really need a platform that can capture the differences in cell states and edit success. So we really need a single cell platform to do this. And this is the sort of data we're starting to look at. This is cell line data, some gene FBXO11 that we're using for methods development. So you can see that if you edit the cells on the right, yes, you have lower expression. Controls, you have higher expression. That makes sense. This is a sort of a loss of function allele that we're putting in. But more importantly, when you look at those edited cells, we can go back and see what genotype we've introduced. And you can see whether you have AA, AG, or GG, we're actually inducing kind of like an EQTL. So depending on how much editing a cell is seen, there's a differential effect on gene expression. So this is a way that we plan to start trying to validate some of these T cell effects. So to summarize this section of the talk, gene regulation can be dynamic across T cell states. The effect of a risk SNP on gene expression may be specific for particular T cell states. A single cell EQTL study that I told you about reveals a broad range of T cell states. These cells can be modeled with continuous variables, which is how we've been approaching it mathematically. And we've been using this approach to find dynamic EQTLs that change across cell states. Strikingly, about a third of EQTLs are dynamic across ex vivo states, and autoimmune disease EQTLs are disproportionately dynamic. this approach may be highly relevant, we think, not just for autoimmune diseases, but for other complex traits and cell types too, because this is a lot of the issues we're dealing with. I think a lot of complex traits are also thinking about. Now I'm going to switch gears and start talking a little bit about the HLA, that big Burj Khalifa tower that I showed you in Dubai for genetics. So now I'm going to transition to talk about the right side of this plot, where I'm going to talk about the MHC and its interaction with the T cell receptor. So how does the HLA interact with an antigen? So for RA, the typical antigen we think about are peptides that are citrulline. We think that that's the driving antigen for most of RA. And when we think about it triggering autoimmunity, we think that the antigen is interacting on two sides of things. So you have an antigen presenting cell, which is using the HLA to present the antigen, and that interaction, it's interacting with the T cell through the T cell receptor. So the antigen specificity of your immune response is coming from two sides, both the HLA and the T cell receptor. And most of us in the world of autoimmunity, we think a lot about the right side here. We think a lot about what we call the peripheral hypothesis. So the idea is you're more susceptible to autoimmunity because you have an HLA risk allele, and that HLA risk allele is really good at presenting a problematic autoantigen to your immune system. We started to think a little bit, is there a role for the central hypothesis? The central hypothesis is where you think about thymic selection, and you wonder if HLA risk alleles are presenting certain thymic peptides and not others, and that's somehow influencing which T cells are surviving to make it to the periphery. So are your HLA risk alleles shaping your T cell repertoire to make you more susceptible to autoimmunity, or perhaps somehow increasing the chance that a T cell receptor that's autoimmune reactive is getting through? The peripheral hypothesis is one that we certainly all believe. There's been a lot of structural studies, for example, this one from Jamie Rossjohn and his colleagues, showing that HLA risk alleles are in fact good at binding citrullinated peptides. And this has been shown for a wide range of autoimmune diseases. But is there evidence for a central hypothesis? And here we're going to talk a lot about the CDR3 region, which is the critical region for antigen binding, which of course defines the identity of a T cell, because the CDR3 region is so random. And we wondered if when the TCR composition is modulated is it modulated in the thymus? Does HLA genotype influence TCR composition? There was already data about V gene selection in humans, which is influenced by HLA, but the alleles driving the HLA alleles driving V gene selection are different from the alleles that drive autoimmunity. So we wanted to ask the question, do HLA alleles influence CDR3 composition specifically? So the strategy that we took was to start looking at TCR sequencing data. There are all these fantastic technologies now to get data on T cell receptor driven by high throughput sequencing. We can infer TCR sequences from bulk RNA-seq data, which is often in the public domain. And then there's also targeted sequencing strategies that let us get TCR sequences from a wide range of cell types. And then what we wanted to do was test the relationship between HLA and TCR. And the idea is if a genetic mechanism of HLA in autoimmunity has a central hypothesis component, then HLA should be associated with TCR features. And for this study, we thought a lot about the CDR3 beta region, which is this highly hypervariable region, again, really specific for autoantigen binding. We looked at two public data sets. So all of this analysis was done in public data. The discovery data set had 666 individuals with targeted TCR sequencing, only TCR beta. They were all HLA typed, and we had about 200,000 T cell receptors per patient. And then we had a replication data set, and it'll become important that these replication T cells were all naive CD4 T cells. And for this data set, it was from RNA-seq data. So we had a much smaller number of TCRs because we were inferring from generic RNA-seq. So we had about 2,000 T cell receptors per patient. So when we looked in this CDR3 region, we saw that and looked for correlations across the positions. We saw that between position 107 to 113, it really looked truly random, which is what we expected in this region, which is what you know, what you would expect to see based on what we know about these regions. And Kaz Ishigaki, who was then a postdoc in my lab and now an independent PI at the RIKEN, started looking at this issue. And basically what he asked is, for a given position like position 110, how many alanines, for example, or pick your amino acid residue, does that person have in all their TCRs? So you would imagine that individual A has 3/10 alanine, individual B has, 3/9, individual B has 6/9. So that gave us a phenotype that we could then ask, you know, is there an HLA association with that? He did a multivariate analysis, and what we found is indicated here. So this is, you know, this is the HLA locus specifically. And what we found is that DRB1 actually had the strongest association in position 13, which was associated with that amino acid composition. So this was really interesting, and you can break it down here and actually look at each of the HLA DRB1 positions, because it's just a protein, and you can look at each of the CDR3 positions here, and what you can see is that there's the central region where about 8 to 10% of the variance of which residues end up in a TCR3 is being driven by that position 13. And this was really exciting to us because in 2012, when we mapped risk alleles for rheumatoid arthritis, we found that position 13 in HLA-DRB1 was the most important position for RA susceptibility. And position 13, along with 71 and 74, were all associated with RA susceptibility. 90% of the signal came from position 13. And those three positions defined a very specific pocket, the P4 pocket for HLA-DRB1, which binds autoantigens. Also in type 1 diabetes, same positions, position 13, position 71, same P4 pocket. So our thinking was that HLA TCR associations are being driven by thymic selection, maybe positive thymic selection, and that different HLA alleles may prefer to bind subsets of thymic peptides, and that might be shifting TCR composition. So where are these CDR3 QTLs coming from? So the first thought was it could be like hardwiring in the genome, like we see in expression QTLs. Could it be antigen experience in the periphery, like as your T cells grow up and experience antigens out there in the wild? Is that what it's about? Or is it really thymic selection? And so to test the hardwiring of the genome, we looked at non-productive CDR3s. So due to sort of mistakes in genetic, recombination to make CDR3s, sometimes you get non-functional CDR3s, non-functional TCRs that are never expressed as a protein, but are present as at the RNA level. So these randomly produced TCRs, sometimes, they have a stop codon or a frame shift. They're never expressed as a protein, and they're not under any selection. So if HLA-CDR3 associations were hardwired in the genome, you'd expect that these non-productive CDR3s at the RNA level would also have an HLA association. And indeed, when we looked at this, we saw that the productive CDR3s, the normal CDR3s, have a really strong association. The non-productive ones don't. So that's telling you that the HLA effect on CDR3 is happening at the protein level, not the RNA level. We thought, could it be antigen experience in the periphery? As you might remember, our replication data set was a data set of naive CD4 T cells, or T cells that had not yet transitioned to become memory T cells. And indeed, when we looked at that, we saw that the associations were actually stronger, suggesting that that what was happening was happening more strongly in naive rather than memory T cells, which again puts you closer to sort of thymic selection. And so that's what we sort of concluded was most likely to be happening. And I just wanted to give you a structural perspective. So if you're looking at position 13, you can see that position is really close to the antigen. It's of all the different amino acid positions, that's the one that gets the closest to the actual to an actual antigen residue. And at the same time, if you look at the CDR3 structure of the T cell receptor, you can see that that's getting really close to the antigen as well. So this is not happening because HLA and TCR are directly interacting. It's happening because of what you see here, which is that position 13 buried deep in HLA-DRB1 is actually pretty close to the antigen, and that antigen is pretty close to the CDR3. So we think it's an indirect relationship. Do risk alleles for autoimmune diseases favor certain TCR sequences? So we took known RA risk alleles, and what we could do is create an HLA risk score. And we could ask what's the TCR profile look like? So for RA, we could come up with what we thought would be a CDR3 that would be favored in terms of thymic selections. We could do the same thing for type 1 and celiac. And then working with our colleagues, Eddie James and Jane Buckner over at the Brenner Institute, we were able to look at citrullonated peptide reactive CDR3s. From the public domain, we were able to look at gliadin reactive TCRs from celiac patients. And what we saw is that those reactive peptides were more likely to fit that profile that we had expected for these different diseases. So that helped us sort of think that probably what's happening is that the HLA alleles are selecting for TCR sequences that are then a little bit more likely to bind an autoantigen. So the possibility that we're thinking about is that HLA disease risk alleles are shaping TCR in the CDR3 regions, and we think that this has implications for autoantigen binding. For autoimmune diseases, HLA risk alleles might be selecting for rare TCRs that may be reactive to autoantigens. And additionally, HLA risk alleles may select for TCRs that skew towards certain states. And that'll be I'll talk about that briefly in the next section. And many TCRs may bind the same autoantigen, but perhaps some TCRs skew towards certain cell fates. So just to summarize this section, HLA alleles influence autoimmune disease risk and also influence TCR composition. Specifically, HLA-DRB1 is associated with specific amino acid substitutions within that CDR3 region. The key driving position, position 13, is the same position that drives the risk of RA, type 1 diabetes, and a host of other autoimmune diseases. TCR amino acid changes track with RA risk effects, and it gives some support for this idea of a central or thymic selection process, and the changes appear to lead to enrichment for very specific TCR sequences. We have some preliminary data that suggests that these TCR sequences appear to be enriched for those binding autoantigens, but of course more studies are needed to confirm. So finally, in this last short section, I'm going to talk about how HLA might be acting on T cell receptor sequences, and T cell receptor sequences might be influencing T cell state. So this is work from Caitlin Lagatuda, who's a current graduate student in the lab. So generally, we think of the TCR as the tool that the T cell uses to recognize antigens, we want to ask the question: can TCR composition actually influence the ultimate state of a T cell? And if so, is it possible that HLA influences T cell states by influencing T cell composition? And this is some exciting work that we have in progress. And we were really inspired by this paper by Chris Garcia in Science about a year ago, where he looked at a specific TCR, TCR55, which recognizes an HIV peptide. And that particular TCR is anergic. It doesn't actually lead to any kind of a T cell activation response. And then he went ahead and he mutated a base pair in CDR3 alpha, and it kept the same antigen specificity, but it made it more likely that the T cell would actually be activated. So that made us think that maybe there are features in certain T cell receptors, in normal non-engineered T cells, which are more likely to be activated, and that might provoke certain T cell states. So this was so Caitlin calls this a cell level genetic study. So here we have a genotype. The genotype is the T cell receptor, and we have a phenotype, which is this complex multidimensional T cell state with large scale RNA-seq profiling. And now what we're doing is we're looking at this complicated sequence of the TCR as our genotype. There's now a lot of public data sets on T cells that are multimodal, where there's both data on the T cell receptor sequence, as well as RNA-seq data on those T cell states. So we've got these 6 data sets with hundreds of thousands of T cells that we're looking at. And the first thing Caitlin did was she took one of these data sets, she annotated the T cells as you see here, and then she started to say, can I find T cells that have the exact same sequence, but from 2 different individuals? So they're not, you can imagine a T cell with the exact same T cell receptor in the same individual might just reflect proliferation. But if you've got the same T cell sequencing in 2 different individuals, they really couldn't have arisen from each other because they're from 2 different individuals. And strikingly, when she looked at this, she found 116 pairs of identical T cell states. And she expected, based on the frequencies of the different T cell states we were looking at, that 25 of them should be concordant, that 25 out of 116 should have the same of the 8 T cell states. In fact, she observed that 81 were concordant. So there was a strong enrichment for a T cell state if you had one of these, if you shared a sequence. So there was sort of a heritability there. So to get a broader view of this, we featurized our TCR sequences. We identified all the variable TCR sequences, and we replaced the residues with some latent variables representing biophysical features, biochemical features such as hydrophobicity and charge and so forth. And then we also took our gene expression data for each of these T cell states, and we applied our old friend canonical correlation analysis to find out which T cell sequence features were correlated with which T cell RNA-seq profiles. And what we found is that the first 4 canonical variants were more correlated than you'd expect by chance. So there were 4 connections. The first one capturing the difference between T cells and NKT mate cells. The second one capturing the difference between CD4 and CD8 T cells. Third one, interestingly, capturing the difference between naive and memory T cells. And the fourth one capturing the difference between regular T cells and regulatory T cells and other T cells. And so now we went through and looked at each of these things, and what we were able to do for each of these different cell state features, we were able to figure out which T cell receptor sequence features promotes which specific fates, as indicated here. We renamed them TCR8 for the NKT mate profile, TCR8 for the features that promote CD8 T cell development instead of CD4 T cell development, CV3, which is really important, which predicts the likelihood of forming memory rather than staying naive, and finally TCR-reg. And we apply this to an external data set just to make sure that we weren't like somehow overfitting our data. And indeed, what we saw is that the score always tracked with the predicted outcome. And I'll draw your attention to that third one, where you can see that the higher your MEM score is, the more likely a T cell is to actually make the transition to memory. The first 2 CVs, the NKT versus mate, that's probably happening because they have a different, they're not interacting with the MHC. CV2 is picking up differences between class 1 and class 2. So those are structurally different. So maybe that's going to promote different T cell receptor sequences. So it's those other 2 that were really surprising. So this third canonical variant, or TCR-MEM, seems to be associated with antigen experience. And indeed, we can see that this is true in both CD4 T cells and CD8 T cells. And naive T cells in general, both CD4 and CD8 on the right, you can see have a much lower score than the memory T cells, which have a much higher score. And this is all kind of ongoing work. Obviously, the next question we were concerned with is like, maybe what we're seeing is just that there's different antigens out there. And maybe this is a score picking up on certain antigens that are common out in the world. But actually, as we've looked across a range of antigens, what we see is that this effect is pretty consistent across all different antigens. So we think that these sequence features are actually predicting sort of a general reactivity, a likelihood of some type of activation, which leads to memory formation. So HLA influences TCR sequence features, and TCR sequence features influence T cell fate. And we identified 4 TCR influenced states that I told you about. TCR features that drive memory formation are similar to those that turn out to drive thymic sequence selection. And what we saw is that the TCR features driving memory formation are somewhat variable across antigen, but consistently have an effect across different antigens. So the possibility that we're starting to think about is that TCR features may be particularly important for subsets of key antigens, for example, self-antigens. And you can imagine a possibility that for a given autoantigen, given self-antigen or infectious antigen, there may be multiple T-cell receptors out there that are present, but only a subset of those have autoimmune potential. And that balance may be influenced by the HLA. So, let me move to conclude. So, how might genetic risk conspire to cause autoimmune diseases? Covered a lot of ground here, but just to summarize, the major risk factor are HLA alleles that present autoantigens, but these same HLA alleles alter TCR composition. And we talked about this idea of a central hypothesis that the HLA risk alleles not only present antigens, but also influence the TCR towards sequences that are both antigen reactive and skews the TCR towards certain pathogenic states. HLA risk alleles are able to bind and present autoantigens in the periphery as well. We also presented data on the diversity of T-cell states and how alleles function to alter gene expression, but in a cell state specific way. The fact that we see allele-specific effects changing across cell states argue that there may be many rare cell states too in which T-cell alleles have large effects. And of course, we think these ideas apply beyond T cells. Non-HLA risk alleles alter expression in critical genes where T cells are in pathogenic states. So it's important for us not just to understand the genes, the target genes, but also the important cell states as we think about how we're going to take this disease on. Future directions, we're thinking about loci outside of the HLA. We're continuing to map effects of regulatory variation across T cells and other immune cells. This requires a lot of computational speedups that we're working on so that we can explore the whole genome. Our current studies have been looking at blood. We think it's really important to think about tissue T cell states because they're different. We're also thinking about the space between genetic variation, the open chromatin, and gene expression. And we're trying to approach this with multimodal RNA ATAC data. And as I told you, we're thinking about functional validation with these single cell CRISPR assays. For the HLA locus, we're thinking about the T cell receptor sequence features and how they predict T cell states. And we want to functionally demonstrate that these predicted risky looking TCRs actually bind autoantigens and are more likely to be activated than not. So finally, I just want to acknowledge a few folks, and then I'll close. So Kaz did a lot of the HLA TCR work. Caitlin has been thinking about the relationship between TCR and T cell state. Aparna has been leading a lot of the work on context-dependent gene regulation. So I want to thank Megan, Samira, Jesse, and Sarah who worked on these projects in general, and also Socios and Salud, who are our partners in Peru, who helped us with the recruitment of these individuals that were a part of this study. And finally, of course, I want to just acknowledge the lab in general. And with that, I can stop and I can take questions.
Questions? I guess I'm interested in how much do you think is really 3D conformational changes to the chromatin in the different states that are affecting EQTLs? You mentioned you're trying to do ATAC-seq, but I mean, do you think you're going to see much, or it's going to be variable, like a T cell that's very far away from this other type of T cell is going to have a lot more conformational shift?
Yeah, it's really interesting. So, one of the things that we're starting to see when we look at ATAC versus RNA data is that the state-to-state variation of ATAC data, of ATAC peaks, is much less.
In the different T cell states, they're not so different from
So in a way, if you look at, this gets a little hand wavy because the ATAC data is like much sparser, as you know, than the RNA data. But what we sort of think we're seeing are superstates, though, when we look at T cells.
Yeah.
So for example, there may be like multiple transcriptomic cytotoxic T cell states, but they look epigenetically pretty similar.
That's
So
interesting.
what we're hoping to see is that the allelic effect at the ATAC level is less dynamic
Yeah.
than the allelic effect at the RNA level, which suggests that it might be the intermediary may be that sort of like a conformational change.
Yeah.
So these are we have it on our mind. We're not doing high C at this point.
Yeah.
But this is what we are thinking about.
You had mentioned that one position 13 and the RA overlap with other autoimmune diseases. I think you said type 1 diabetes. And does that also extend to like, lupus and our psoriatic arthritis, or do you see clustering of autoimmune diseases that go with particular genotypes? I guess.
So largely, so the answer is like yes, no, and also maybe. For some of these diseases, we've been able to map HLA really effectively. Some of them we haven't been. A lot of the diseases that are autoantibody positive, like RA and type 1, they tend to be on the class 2 side of things. The antibody negative ones, like psoriasis, psoriatic arthritis, ankylosing spondylitis, they tend to be more on the class 1 side. Of the diseases that are on class 2, for RA and type 1 is interesting because the risk is kind of shared between DQB1, which we think is actually likely to be a regulatory effect, as well as DRB1. So it's like both genes. Position 13 is the main driver. Position 13 has emerged in some lymphomas. It's emerged for vasculitis. So it's come up in a bunch of different diseases. What's also interesting is, in position 13, I mean, only in the HLA, right? So you can have 6 different residues at position 13, which suggests that has been under a lot of positive selection. So it's somehow important. And what else is interesting is a residue which is driving risk for one of these diseases might be protective for another,
That's right.
because you have, different families of antigens
that
are the problematic antigens for these different diseases.
Right. Makes sense. Other questions? Are there any online?
There.
Yeah.
How you came to the study population in Peru?
Yeah. So our lab spends a lot of its time studying non-Peruvians, but for this particular study, so it's an interesting story. What that study was actually originally what we did it for was to understand TB susceptibility. So the individuals we recruited there were individuals who didn't have TB but had a history of having TB. So we were looking at them about 5 years or more after they had TB. And so that's why the work was done with Socios and Salud, which is Partners in Health, in Peru, which is focused on stamping out TB. And it's kind of interesting. What we found was that the people who had a history of having TB, so we got into this because we knew that, T cell suppression, for example, or T cell elimination, for example, with HIV, right, causes people to reactivate TB. So we hypothesized there must be some T cell state that maybe is a little bit more likely to be deficient in individuals who are more likely to do badly when they get infected with TB. And that's what we found. We found that even looking 5 years later, that individuals who had progressed to TB, they on average had fewer of this particular Th1, Th17 T cell state that others have now found actually in the lungs of individuals who are affected with TB. And we can't go back and look at people before they got TB, unfortunately, in our current state of time travel. But we did find bulk RNA-seq data that was serially collected in individuals from when they got exposed to when they got sick. And the people who got exposed and got sick had signatures of this T cell state that was lower than individuals who didn't. So there's a whole nother kind of interesting thread in the lab that we're interested in, which is trying to understand the antigen specificity of this infectious disease, looking at these T cell states.
Technical question for you. The part where you talk about PKL association within different cell states. So did you extract PKL data from a single cell, or did you separately profile the genome of those data?
So all of those individuals were whole genome genotyped. So we ran a GWAS array on all of them. We have not been so bold to try to call genotypes from single cell data. I think probably to some extent you can. We've thought about it, because you're picking up, that particular study. It's a 3' based sequencing study. And every now and then this does come up because there's a lot of wonderful single cell data sets out there without genotype data. So we've often wondered, like, can we get that data, look at the raw reads and call SNPs from those data sets? And long story short, I don't think that we think we could do that very well. Again, because you're not even picking up the whole of the transcript, really. You're just picking up this kind of tail end of the transcript. So it's not really enough to impute anything in a reasonable way.
I think you're available for more questions, but let's do it outside where there's some food and beverage and we can all enjoy that. So let's thank our speaker. And please join us outside.